Hello, everyone. Well, if you are in the northeast of the US, it's certainly time to take out your summer clothes. I'm going to see how long I can last without having the AC on because I know that will create a lot of background noise. Um, so we'll see how that goes. Today, we're going to be covering transcription and mRNA processing, particularly in eukaryotes. We'll see if we could fit in some prokaryotic stuff, but there's a lot to cover in transcription. So I really would like everyone to focus and go back into the book and make sure that you know all the steps for transcription, because this is a really important process that is, of course, part of the central dogma. Now, I know that we had our exam yesterday, and I'm sure that some of you have handed in the essays today, so great job on completing that. I will be posting the multiple choice grades after I finish this lecture. I did go through everything yesterday and make sure that everything was graded evenly and that there were no bugs in the multiple choice grading due to, the automatic grading. And so I just wanted you guys to know that I do go through that and look at things, maybe not every single question for every single person, but if I find an error in grading for one person, then of course I go back through and look at it for everyone's grades. But my point here was to say, I know that we just had an exam and I'm sure that everyone was studying a great deal, but you really have to keep up the work, keep studying, keep reading ahead, especially because we're going to be going through very important topics the rest of this week. So transcription. Let's just review quickly the structural differences between DNA and RNA. So we see here that we have RNA and DNA and the sugars in them. So as we discussed, DNA is called deoxyribonucleic acid, but this right here is labeled deoxyribose because this is only the sugar, the ribose. And here on the left side, we have RNA, and this has the additional oxygen right here, and that's only on one carbon. And just remember that the other carbon at the bottom here is the 3' carbon, and that one is present, the oxygen is present in both RNA and DNA, and that's the one that is needed in order for a nucleotide to be added onto the strand, the growing strand of DNA. And so the sugar is part of the backbone of DNA or RNA. And we see here the other part of the backbone is the phosphate group. And again, we have the phosphodiester bonds, which hold together a single strand, the nucleotides in a single strand of DNA. And then we have, again, hydrogen bonding that would be present in DNA and sometimes in RNA. And that holds together the two strands of a DNA helix or double-stranded RNA. Again, that's hydrogen bonds hold together two strands of nucleic acid. Phosphodiester bonds hold together the nucleotides within a single strand in either DNA or RNA. And as I'm sure all of us have heard at some point before, we have some differences in the bases, or rather just one difference in the bases used between RNA and DNA. So DNA uses thymine, and so that's the T, whereas RNA has uracil. So we have uracil instead of thymine in RNA, and then in DNA we have thymine but not uracil. So if you're looking at a DNA, or rather, if you're looking at a nucleic acid sequence, and that sequence contains U's, then you know it must be RNA. If it contains T's, then you know it must be DNA. And the uracil that we see here and the thymine, you can see that their structure is pretty similar. So they both pair with the A's, the adenines, in a double-stranded form of nucleic acid. And that's why they're interchangeable, basically, between DNA and RNA. Okay. So transcription in a nutshell. So here I'm just going to go through all the steps as an overview so that when I go through them in detail, you can know where we're going. So step one is binding and unwinding, very similar to what we saw in DNA replication. Step two is the initiation of synthesis. So similar to DNA replication, we need to actually start making the new molecule. Then we have elongation, which is adding on monomers, and then termination, which is the end of making that new molecule. And this is the one that we really didn't talk too much about in DNA replication. So again, we have two strands of DNA. And when we talked about DNA replication, both of the strands were going to be copied. So both strands end up being the template strand. However, in transcription, this is not the case. Both strands of DNA are used at some point in the lifetime of a cell. But in certain areas, at a certain specific locus for a certain gene, only one strand is going to be used by RNA polymerase to carry out transcription, to express that gene. So let me say that one more time. You have two strands of DNA. Both of them, over the course of the entire DNA helix, are used at some point for transcription. But when you're thinking about a single gene, so if you're thinking about that eye color gene for the fly or the flower color gene for the peas, you only see the use of one strand being transcribed. And it's always the same strand whenever that particular locus or that particular gene is used, is expressed, is transcribed. And here we see, and this is just completely arbitrary, it's not always the bottom strand, but this is the way it's set up in this image, that the bottom strand is the template strand. So the template strand is the one that is read by RNA polymerase. The other strand, the complementary strand to the template strand, is the coding strand. Then in the middle here, we see the mRNA, which is being created. So similar to what we saw with DNA synthesis or DNA replication, the mRNA, the new nucleic acid, is created 5' to 3'. So that is the 5' end, or the permanent 5' end is created first, and then the 3' end keeps changing as we add new nucleotides. And similar to DNA replication, the template strand is read in the opposite direction. So the RNA polymerase is moving from this end, the 3' end, and it's moving in this direction towards the 5' end. So if you get confused, which believe me, everybody does get confused at some point about this concept, try and draw it out. Draw both strands of the DNA, draw the direction of the polymerase, and then you can draw the new strand of mRNA. And that will help you figure out what is 3', what is 5', what direction everything is moving in. Okay. Yes, and so one other thing I wanted to point out here is that the mRNA is complementary to the template strand. The coding strand is also complementary to the template strand, which means that the coding strand and the mRNA have the exact same sequence, except the U's are in place of the T's. And if you want to prove this to yourself, you can look at the template strand. The last nucleotide that was, or the last two nucleotides that were added here, they are an A and a C here. And then on the mRNA, we have a U and a C, which are U and a G, which is complementary to the AC. And then here on the coding strand, instead of the U, we have the T, and we also have the G. So again, the coding strand is the exact same sequence, except for switching out the T's and U's, as the mRNA, and then they are both complementary to the template strand. All right, and we already talked about this. And as I alluded on the previous slide, either strand of DNA can contain the RNA template. It depends on what gene you're talking about. And preceding every single gene, in every single case, there is some kind of regulatory sequence. And this is called a promoter in prokaryotes. And so we see that there is a promoter sequence here, which is upstream of gene A. So by identifying where the promoter is, then you could identify which strand of the DNA contains the template. Because, or rather, as long as you know what the 5' and the 3' strands are. So if you take a look at this image here, we see the promoter. We know that the RNA polymerase is going to start at this end and move towards this end. So it's moving from left and going towards the right. And so then which strand is the template strand? I'll give you a minute to try and think about it. So we know that the polymerase is going to read 3' to 5', which means that the bottom strand is the template strand for this particular gene, gene A. If we take a look at gene B, the promoter is on this end, and we know then that the RNA polymerase is going to be moving from the right side and moving towards the left side. And just from that, comparing it to gene A, we know that it's going to be using the other strand as the template strand. But if you didn't have gene A to reference, then you could go through this again and say, okay, it's moving from right to left, and we know that it's going to read 3' to 5', so we start at 3', it has to be the top strand. All right. And you can see evidence of either or both strands rather being used if you go into the NCBI website, which is where you could find many genomes. And if you look at a genome, you can see which strand of DNA a particular gene is transcribed from. And so this, this image here shows a region of the human genome, has 2 genes, and the 2 genes are read on opposite strands or different strands of DNA. And you can see arrows pointing in opposite directions. So here we see an arrow pointing this way, and then here this arrow, it's a little bit hard to see, but you zoom in, this arrow is pointing towards the right, and the other arrow here is pointing towards the left. So these guys are coded for on different strands of the DNA, even though they're very close to one another, relatively close to one another in the genome. Okay. So let's talk about the first step, which is binding and unwinding. So there are lots of genes in the genome. How is it that an RNA polymerase is going to bind to one area and successfully transcribe that gene? How does it know to bind in the right place and not bind in the middle of a gene? Well, that is due to this control region. And I mentioned this on the previous slide. This control region in prokaryotes is called a promoter. It's also referred to as the negative 35, negative 10 sequences. And that is because 10 base pairs or 10 letters upstream, and that is before the actual gene, we have these sequences which the RNA polymerase recognizes and binds to. So here we have the negative 35, here we have a negative 10. We see that there are a lot of A's and T's in these sequences. So these sequences are not consistent, they're not identical between different genes and different species, different organisms, but they're similar. You get recognizable promoters, are negative 35 and negative 10 sequences in the promoter. And these sequences are always upstream of the start site. So let's go back to this image for one moment. So what does it mean to be upstream? It means that when the RNA polymerase is reading from 3' to 5', that promoter is going to be closer to the 3', and then the gene in comparison is going to be closer to the 5' end. So the promoter here is upstream of the gene if you're moving in this direction. And then for the other gene, where we know the RNA polymerase is moving from right to left, then the promoter is on this side. It's still closer to the 3' end, and then the gene is closer to the 5' end. So that's what we mean by upstream. Then downstream would mean the opposite. So the gene is downstream of the promoter in all cases. All right. So the promoter is not actually transcribed. No RNA is made from the promoter. This is just a place where the RNA polymerase, this protein, is going to bind. Then the place where transcription actually starts is where we say that the gene actually begins, and that is here at the +1, the start site. So actually, I'm not going to go through this. You should recall this. We did go over this in one of the first lectures, what a prokaryote is. Long story short, it's a bacterium or an archaeocell. And it does not include eukaryotes, which are all multicellular organisms, and they include things like mammals and humans. All right, so prokaryotic RNA synthesis. RNA polymerase binds. It binds to the specific area because of a sigma factor. So a sigma factor is another protein. A sigma factor is only used in prokaryotes, and the sigma factor helps the RNA polymerase find that promoter. So without the sigma factor, the RNA polymerase wouldn't be able to find the promoter. It would just kind of be wandering around on the DNA. So because of the sigma factor, which is represented by this yellow thing here, the RNA polymerase can bind in the correct spot. And so we see here again that we have the sigma factor, which is bound right at the start of the promoter. So that would be the -35 sequence. And then we have the RNA polymerase binding over the sigma factor or with the sigma factor. And the sigma factor kind of lines up the RNA polymerase so that the sigma factor, even though here it's shown as symmetrical, it's better if you think of it as like a little triangle, a little wedge of cheese if you go with the color scheme here, pointing in this direction towards, oh, I have the mirror on my video, so don't pay attention to what I've just pointed to. It's going towards the right side here. And that sigma factor is asymmetrical, and then the RNA polymerase is also asymmetrical, so it always binds pointing in the right direction. With the -35 sequence, the back of the RNA polymerase, and then the -10 sequence is closer to the front of the RNA polymerase. So that the RNA polymerase is facing, so to speak, the gene to be transcribed. And I think that I was really fixated on this one phrase in the textbook. I don't know if they even kept it in the present edition, but it said that RNA polymerase slides down the DNA until it hits a promoter. So basically, RNA polymerase is going to bind the DNA like wherever, and then the sigma factor is going to tell the RNA polymerase, stop. This is a gene where we need to transcribe. And so I just had this little GIF, which is actually kind of miserable for the cat, but that RNA polymerase is sliding down the DNA, and the only thing that puts the brakes on, again, is that sigma factor when it hits the -35, -10 sequence. Okay, so as we said, the sigma factor recognizes promoter. Then the RNA polymerase changes conformation. It binds the DNA more tightly, so it can't just zoom down the DNA anymore. Now it's actually going to clamp down, and it's going to start reading the DNA and transcribing. And so this is when synthesis begins. Also, as the RNA polymerase tightens the binding, as that conformational change happens, the sigma factor leaves. The RNA polymerase cannot bind tightly if the sigma factor is present. All right, so now we've gotten through kind of the initiation of the transcription. Now we'll move into the actual synthesis of mRNA. So beginning that synthesis and then elongation of the mRNA. So because this is RNA, similar to the case with the DNA replication using the primase, which sets down the RNA instead of DNA, in this case, we don't need free 3' hydroxyl in order to lay down the first base. And so we can just start transcribing, start creating a strand of mRNA without needing to add on to something. Again, because this is RNA instead of DNA. So initiation is easy. Then elongation occurs through this process, which is shown here. We have incoming ribonucleoside triphosphates, so very similar to what we saw with DNA replication. We see these nucleotides. They have 3 phosphate groups instead of a single phosphate group, and 2 of them are going to be removed. And these guys come in through this triphosphate tunnel, which is just a structural feature of the RNA polymerase. And then we have the active site of the polymerase, which is where the bonds are actually formed. So again, that is the phosphodiester bonds between the growing strand and the incoming ribonucleosides. And this is also where the hydrogen bonds are going to form spontaneously. And then here at the back, we see that the hydrogen bonds are broken between the mRNA and the template strand. And the 5' end of the mRNA is going to stick out into the cytoplasm. Because again, let's recall, in prokaryotes, the cells do not have a nucleus. So all of this is occurring in the cytoplasm, just in the general area inside of the cell. And I do want to just point out again the active site here, because we haven't talked too much about active sites in the past, but all enzymes have at least one active site, and that is the region of the protein where the reaction, the chemical reaction actually occurs. Bonds are broken and or formed. And in the active sites of enzymes, they have a very particular structure, and if that structure changes at all, oftentimes you're going to lose the function of the protein. So if you see a mutation in a protein and you know that mutation is in the active site, then it's fairly likely that that mutation is going to cause the protein to be non-functional. And so elongation, that is what we just talked about on the previous slide. And again, we are making RNA, or particularly mRNA. And we are moving over the gene, and we are approaching this red area. And that is where the synthesis is going to end. So the termination sequence is this red sequence here. So, well, it was labeled as the red sequence, or the terminator here, even though it shows here the RNA polymerase is not quite at the termination sequence yet. But when it gets to this red part, it's going to hit the sequence that causes the release of the polymerase and the mRNA transcript. And that is just a characteristic of this particular sequence. It's going to be difficult for the polymerase to bind to and to move past. And so basically it stops synthesis because it kicks off the RNA polymerase, or at least stops it from binding tightly. And so that's the end of making this piece of mRNA. And this is also characteristic, it's unique to only prokaryotes. Eukaryotes have a different way of ending the transcription. So here we also see typical transcriptional terminator sequence, and after that terminator sequence is the stop site. And then we see here that the sigma factor can rebind to the RNA polymerase after it has been kicked off the DNA, and they could go ahead and find a new gene to transcribe, or they could again transcribe the same gene. So if we didn't have that terminator sequence, then we would continue to transcribe, and we're going to see in a moment why that is relevant. So we went over this already. Okay, sorry about that. So I needed to take a minute because, can you believe it? I didn't have a slide for this. I think I always used to draw it on the board. So we've been talking about prokaryotes, and prokaryotes have this neat system where they can transcribe or express all these genes needed for a single pathway or a single function at the same time. And they do this by transcribing all of these genes on a single piece of mRNA. So down here we have a piece of artificial DNA. That's why it says expression vector. And we see this DNA has a promoter, and then it has gene A, gene B, gene C, and gene D, and then it has a terminator. So even though we have 4 genes here, we only have 1 promoter, we only have 1 terminator, and that means that the RNA polymerase is going to start transcribing here at the promoter. It is going to transcribe all 4 of the genes at once in one single piece of mRNA, and then it's going to end transcription. And then you're going to get the synthesis of all 4 of these proteins from this single mRNA. And so this is called polycistronic mRNA. And so polycistronic mRNAs contain several genes. They're often part of, or they often come from what is called an operon. So we'll talk about that in a couple minutes, or in another class rather. And we'll say here, they do not have operons. And operons are just sets of genes that are expressed together. So eukaryotes do not have operons, and therefore they do not have polycistronic mRNA. Prokaryotes do have these operons, or these sets of genes that are expressed together as one single polycistronic mRNA. All right, and then when you have that polycistronic mRNA, we can see that there are multiple gene sequences on it, and each one can result in a protein. We'll come back to this slide in a couple of minutes, or rather tomorrow. So don't don't get confused by it right now. All right, so moving on to eukaryotes, and I have this special seal that I made to make sure that we don't confuse the prokaryotic processes with the eukaryotic processes. So eukaryotic genes are set up a little bit differently than prokaryotic genes. So they have a TATA box upstream of the gene. And so this is where the RNA polymerase is going to bind to the DNA upstream of the gene. The TATA boxes contain T's and A's. So again, we come to that theme of T's and A's, those pairs having fewer hydrogen bonds and representing a weaker area of the double helix. But in addition to that upstream sequence of the gene, there are many other regulatory sites that are unique to different species or different groups of organisms, and they will bind to different proteins that are not RNA polymerase that will affect where RNA polymerase binds to and what genes are eventually expressed. And we'll talk more about this in the future, and we'll give a hint at it today. This, as you can see, eukaryotes, I always call them the divas. Like, they're so complicated, they require so many different proteins that, you know, bacteria are nice and easy. But it's okay, because I guess we are eukaryotes, so we shouldn't put them down too much. So eukaryotes have 3 different RNA polymerases, and the 3 different RNA polymerases are used to transcribe different sorts of genes, or different sorts of RNA, rather. So RNA polymerase 2 is the one that is used for mRNA, so that's most of the genes that are being transcribed. And then RNA polymerase 1 makes ribosomal RNA, so that is a sequence that never becomes protein, but it makes the ribosome. And then RNA polymerase 3 codes for tRNA, that is transfer RNA, and some other types of small RNAs. So when I refer to RNA polymerase and then I don't specify which one, assume that it's RNA polymerase 2. So the initiation of transcription in eukaryotes is much more complicated and requires many more proteins than prokaryotes. We're not going to memorize every single protein that is needed because that would be pretty pointless. We need to save your brain space for other things. I'm gonna make this a little bit smaller. But the name for all of these different proteins that are needed to bind upstream of the actual gene to facilitate the initiation of transcription are called general transcription factors, or GTF. And these general transcription factors are a set of proteins that are needed for the initiation of transcription, and they assemble on a promoter sequence, which again is going to be upstream of the gene. And the promoter, or part of the promoter, is the ta-ta box. So if you see here, that's the green sequence, and we see this first protein bind to this area. So this first protein, this very first general transcription factor, is called TF2D, transcription factor 2D, and it contains an area called the TBP. And if you kind of imagine what the possibilities for what that stands for, it's the ta-ta binding protein. And so the TF2D is a complex of multiple proteins in itself, and one of them is the ta-ta binding protein, which allows it to bind upstream of the gene. And so if this were prokaryotes, we'd be pretty much done. Like, okay, we've got something that's oriented in the right way, upstream of the gene, like, let's go. But in eukaryotes, that is not the case. So we need many other general transcription factors in order to begin transcription in eukaryotes. And so we have this next transcription factor called TF2B. We also have TF2E, TF2H, TF2F. There are lots of these. Some of them will bind to the DNA, some of them will bind to other general transcription factors. This one here is very important, TF2F. So that is going to actually phosphorylate the RNA polymerase 2. Sorry guys, so let me correct myself. So TF2H is the one that phosphorylates RNA polymerase 2. TF2F is phosphorylated, but that's not so important. The takeaway here being that RNA polymerase 2 can get to the promoter, it can bind to the DNA, but it's not going to actually start transcription until it is phosphorylated. So it's quite important. In addition, I want to point out that some of these other transcription factors that are part of the general transcription factors for initiation of transcription, they are going to be important for interacting with the mRNA as it is produced. All right, so again, we have various proteins arranging themselves on the DNA, near the DNA. We have RNA polymerase II, and then we are going to have the phosphorylation of RNA polymerase II, which by TF2H, which I wrote it correctly here. And we see all these organized here, and then eventually, once the RNA polymerase is phosphorylated, it is going to start transcribing, and then all of these other general transcription factors are going to leave, except for the ones that are going to interact with the mRNA as it is produced, and these are the ones that are actually bound to RNA polymerase rather than the DNA. And here at the bottom, in the bottom image, we can see the phosphate groups that were added to the RNA polymerase. All right, and so this RNA polymerase works very similar to the RNA polymerase in prokaryotes, so it's going to take in these triphosphate ribonucleosides, and then it's going to remove two of the phosphate groups, going to match them up to the template strand and form the phosphodiester bond. And then as the mRNA leaves the RNA polymerase, we have the breaking of hydrogen bonds between the mRNA and the template strand of DNA, and then it's going to be available here where it's going to be worked on by some of these other transcription factors that we won't talk too much about in detail right now. So as RNA polymerase transcribes, mRNA is being processed. So many of you have probably heard about splicing, the concept of splicing introns and exons. And that and a number of other things have to occur before the mRNA can actually be translated. And so I just have this cartoon where the processing enzymes say to the unprocessed mRNA, "You're not going out of the nucleus like that, are you, dear?" And that is because the unprocessed mRNA literally cannot leave the nucleus because it doesn't have the appropriate structural features that will get it through those nuclear pores. So what occurs here, or what occurs next, are capping at the 5' end, splicing throughout the mRNA, and then the polyadenylation at the 3' end of the mRNA. So we see here that the RNA polymerase II has these different general transcription factors here that we talked about. Some of them are capping factors, some of them are splicing factors, and some of them are polyadenylation factors. Here we see that there's one of each. That varies depending on the organism that you're talking about, but for our purposes, this is fine. And you can see that they are attached to this tail of RNA polymerase, which does not exist in the prokaryotic version of RNA polymerase. And they are attached to the tail so that they're ready to kind of jump in and modify the mRNA as it is being made. They do not wait for the entire mRNA to be created or synthesized before they start modifying it. And the result of the processing of these different factors is that we have a 5' cap added. So obviously this is at the 5' end of the mRNA, and this 5' cap is just a modified nucleotide. And so it is guanosine, which has 3 phosphates added to it. And so normally we wouldn't see the 3 phosphates, we would see 1 phosphate in the sugar phosphate backbone, but here we see 3. And this also has a positive charge to it, so it's a little bit, it's not a normal G that you would add. And then, in addition, we have a poly-A tail, and the poly-A tail is just a series of A's at the end of the mRNA. Notice here, in addition, that we have some non-coding sequence, both at the end between the coding sequence and the poly A tail, and at the beginning, so between the 5' cap, which is like a little hat, and the the coding sequence here. So the bright blue is non-coding, the dark blue is coding. Only the dark blue is going to result in protein. All this other stuff, the 5' cap, the non-coding sequence here, the non-coding sequence here, and the poly A tail, none of that is being read by the ribosome. None of it is going to be translated. None of it codes for amino acids, or none of it should code for amino acids. So, as I said a moment ago, the unprocessed mRNA is not structurally fit for leaving the nucleus. However, as this 5' cap is added and the poly A tail, that is going to prep it to be able to leave the nucleus, because the 5' cap, as well as the poly A tail, and also certain areas within the coding sequence, are going to be bound by these other proteins in the nucleus, and these proteins are going to assist the mRNA in getting out of the nucleus through one of those nuclear pores. So the 5' cap is only used for getting, more or less only used for getting the mRNA out of the nucleus. The poly A tail also protects the mRNA, so it has this additional function even after it leaves the nucleus. And then we'll talk about splicing now. So splicing is when certain areas of the gene are removed from the mRNA. And there are different regions of an mRNA. This only in eukaryotes, so this does not exist in prokaryotes. There are exons, which are coding regions, and then there are introns, which are non-coding regions. So the exons are going to be kept. The introns are removed. And the process through which the exons are kept and the introns are removed is this process called splicing. And so in splicing, the phosphodiester backbone is going to be broken and then religated after certain areas of sequence are removed. And so here we're actually seeing the gene, that's why it's double-stranded, but these sequences are also going to exist on the mRNA, on the unprocessed mRNA. And so this splicing is actually a pretty complicated process. We don't need to know all of it, but you should know, again, which areas are kept and which are removed, and that the spliceosome is the aggregation of proteins which are formed by these SNRNPs. And when these SNRNPs form the spliceosome, that is what recognizes the area that need to be removed, the introns, and also breaks the phosphodiester backbone so that that is removed, and so it puts the intron into this little circular shape so that it's not mistaken for a piece of mRNA that should be capped and polyadenylated, and then the spliceosome dissociates. And so this is what, I believe this is what is shown in your book. And then this mature mRNA, so this already has the 5' cap and the poly A tail over here. And again, we see that you could call the before version of the mRNA the unprocessed version, or the pre-mRNA, the immature mRNA. Any of those just refers to this mRNA that doesn't have everything done to it yet. So it hasn't been spliced, maybe it doesn't have a 5' cap, maybe it doesn't have a 3' end. But the 5' cap is added initially, because if we go back to this, or rather, that doesn't really show it. This doesn't show it either, but this is the 5' end, so that's going to be made first, and that allows the 5' cap to be added before the rest of the mRNA is transcribed. So naturally, the 5' cap is added first, and then the 3' polyadenylation can't be done until the entire gene, the entire sequence has been transcribed. So this is another view of splicing, and it's shown a little bit differently. So here we're just seeing the snRNPs, and here we get to see inside of the snRNPs. And we can particularly see that there are these sequences in snRNPs. So snRNP stands for small nuclear ribonuclear proteins. I know that's a lot, but if you remember this, if you can remember this, then you will remember that these guys are proteins, which allows them to have this enzymatic activity and break the phosphodiester backbone. And they also contain RNA fragments. So that's the key part. They have the protein, and that is what actually does the cutting and the ligating of the phosphodiester bond. But then the RNA portion is really important because that allows these snRPs or SNRPs to locate the appropriate regions of the mRNA and not just cut anywhere on it. So the SNRPs find these sequences that are unique to the ends of the introns, so the beginning and the end of the introns. And they do this, they recognize this because of these portions of RNA inside of them, and then the protein part is what is going to actually do the cutting. So my cat Elsa has just joined us because I think she wants to learn about transcription as well. And the complete spliceosome contains these SNRPs, which also contain these RNA fragments. So I know it gets quite complicated here. But that whole process is how these introns are removed, and then the exons are ligated together. Finally, here we see that introns have these specific sequences, again, which are recognized by this RNA, but they're highly variable, and that's probably for an important reason, because when we have these genes, these eukaryotic genes that have many different exons and introns, they can be mixed and matched. So all of this is going to be transcribed. All of it is going to be in the pre-mRNA or the immature mRNA. But then which exons are kept and which ones are spliced out is variable. So the introns are always removed. The exons, sometimes all of them might be kept in, sometimes only a few of them, sometimes the first half of them, sometimes the first three, or sometimes the last three. It depends on the expression, the protein that is needed in the cell at that point. And we'll talk more about how this is decided in our problem session. Okay. So let's talk a little bit more about how the mRNA leaves the nucleus. So again, it leaves the nucleus through these nuclear pores. It is large enough to be hindered by these nuclear pores, but it is able to get out because of certain proteins that bind to the mRNA. And this is just kind of a neat structural simulation of the interior of a nuclear pore, where we have all these unstructured regions of peptides. And you can see how they really kind of clog up the inside of the pore, and larger molecules are not going to be able to get through that just by dissolving through. All right, and so here we see the mature mRNA, which has the 5' cap and the 3' poly A tail. It's also been spliced appropriately, and there are proteins that recognize the 5' cap, the poly A tail, and regions that have been spliced. And so all three of these types of proteins are important for helping the mRNA leave the nucleus. And so that they help to push aside all of these long, unstructured regions inside of the nuclear pore. So they kind of push them aside so that the mRNA can get through, and then it's going to end up in the cytosol. And in the cytosol, we have an exchange. The protein that once bound 5' cap leaves, and then we get a different protein binding that 5' cap, which is needed for translation. Sorry, the camera was blocking that. So finally, we see, when we look at this from the big picture, that we have control over the amount of protein that is produced right from the get-go. So we can control how many mRNA transcripts are made, how many times this template strand of DNA is actually copied, and then we could control, which we'll talk about tomorrow, how much of those are translated or how many times they're translated into protein. And so you could have very different numbers of protein from gene A and protein gene B because of these different forms of control. So again, you could make more or fewer copies of the mRNA. You could also splice them differently. That's not going to change the number, but that's also a form of control. And then, again, we can also translate them a different number of times. So that covers transcription. I have a slide here at the end which shows all of the important, or most of the important molecules. So make sure that you're familiar with all these words and concepts. And I'll see you guys tomorrow. So I will be posting the grades later today. And in addition, I'm going to be posting some homework for this.